Hi, everyone. Welcome to the cerebellar seminars. Today we have Brandon Stell. Brandon studied biology at University of Colorado, where he looked at swimming and biomechanics of swimming. He built a robotic leech to measure forces that take place during swimming motion. Then he went to UCLA in Los Angeles to to get his PhD, study his neuroscience with Esteban Modi, who worked on the effects of GABA receptors found outside of the synapses in the hippocampal neurons. Then in 2005, he went to Europe. He moved to Paris. And he started researching on GABA receptors in the cerebellum as a postdoc with Alain Marty. And in 2014, Brandon became an investigator at CNRS, where he's now leading a team that uses in vivo imaging and electrophysiology to study the cerebellum during goal-directed behaviors. And he's going to talk to us today about error signals in declining fibers during motor learning. Brandon, thank you so much.
Thank you, Reza, for the invitation and for, of course, putting together this great seminar series. I'm going to talk today about this new behavior that we developed that allows us to look at climbing fibers in a sensorimotor adaptation task. A textbook example of cerebellar learning is this prism goggles adaptation task that we are all in the cerebellar field familiar with. In this case, a subject aims at a target, throws a clay ball at a target, and they record the position of their throws. And a healthy subject hits the target pretty accurately. They then put on prism goggles, which shift the field of view off to the side, and the subject then misses to the side of the target when throwing the clay ball, but then quickly adapts. Within just a few throws, few tens of throws, they start hitting the target again. They remove the goggles and then they hit to the opposite side, and within a few throws, start hitting the target again. And we got really interested in this phenomenon or this observation, like a lot of people in cerebellar neuroscience. And two things, well, I mean, it was shown by Martin et al. In these classic papers that it requires the cerebellum. And we were really interested in how this can happen in so few trials, in just tens of trials, when most cerebellar tasks are taking hundreds, if not thousands of trials to make an adaptation. So we wanted to study this, and we had to create a new behavior. We had experience recording from Purkinje cells in lobule 4-5 of the cerebellum on the surface of lobule 4-5. So we wanted to have a behavior that engaged lobule 4-5 and would allow us to take advantage of our electrophysiology and imaging skills to observe the cerebellum and Purkinje cells during the adaptation. In these classic stainings from Raugrok in 2008, he shows that when he puts rabies into a limb muscle and looks at where the rabies goes in the cerebellum, he sees these bands of Purkinje cell staining, including on the surface of lobule 4-5. So we thought, okay, let's see if we can use create a behavior that engages lobule 4-5. By the way, feel free to interrupt me at any time. That's totally fine. So to create this behavior, there were a bunch of joystick behaviors, and we thought, okay, let's try to create a joystick behavior where the mouse adapts quickly. After several different goes at different behaviors, we finally converged on this behavior here, which is very simple. A joystick pops up, and like most of the other joystick tasks, the mouse grabs it and moves the joystick to its face. The difference here is that the joystick is controlling the position of the lick port. So when he grabs the joystick, he moves the lick port, and he has to bring the lick port to its mouth in order to receive the reward. And mice learn this task extremely quickly. So the first day that we put the mouse on the setup, they will perform the task tens, if not hundreds of times. So the mouse has never seen the setup before. We put him on, and he kind of, eventually bumps into the joystick with one of its paws and realizes that it's connected to this lick port, and then will systematically bring it to its mouth to receive the rewards. And in about a week, they will do 1,000 pulls in an hour. So it seems to be pretty robust and pretty repeatable. The way we make it an adaptation task is we change the link between the joystick and the lick port. So the position of the lick port is then twice the position of the joystick. So this is kind of like if you ever have worked on a new computer and the mouse is calibrated slightly different and the cursor moves much faster on the screen than you're used to, but very quickly you get used to it. This is the same sort of thing for the mouse. So exact same task, it's just that the lick port is now, the position is twice the position of the joystick, and he has to adapt to that new configuration. So like I said, the mice will perform this. They love doing it in an hour. So this is a plot of the cumulative number of times they will pull on the joystick versus time. In an hour, they'll pull anywhere from 400 to 1,000 pulls on the joystick. This allows us then to change this connection between the joystick and the lick port systematically. We can increase the gain for 5 pulls, then decrease it for 5 pulls, and just switch back and forth 50 times to introduce this perturbation over and over again within the same session. We're of course tracking the position of the joystick and of the lick port at the same time. Throughout the talk, blue is going to be the position of the joystick, orange is the position of the lick port. We see here that the mouse grabs the joystick in its home position, brings it to its mouth here, shown in gray. In this particular set of pulls, this is the first time he pulls in the new gain condition when the gain has gone up. So he pulls the joystick to his mouth, but the joystick is past its mouth, and he has to make a correction to bring the lick port back to its mouth to receive the reward. And because we have the position, we can calculate many different things, including the velocity and the acceleration. The acceleration, of course, being proportional to the net force on the joystick. And if we look at the net force on the joystick, we see there's this point, as the mouse is bringing the joystick towards its mouth, it eventually has to slow down in order to get it to stop in front of its mouth. And this point where it slows down has a position. So at some point between the home position and its mouth, there's this point where there's no acceleration. And we thought maybe that's a good point to kind of quantify each pull, because it's proportional to the force the mouse is putting on the joystick. And also it happens in kind of, not the planning stage, but the execution stage before he reaches his mouth. And so we thought this might have something to do with the motor plan that the mouse is trying to execute. To convince ourselves of that, what we do is we compare the last pull in the gain down situation, so in the control or the recovery situation, to the very next pull in the situation where the gain is now increased and the lick port is moving faster. So this is the trajectory of the lick port in those conditions. And you can see at the beginning, those two pulls start out exactly the same. And then there's a point where they start diverging. And if we do the subtraction of those two positions, we can see where they start to diverge. And that position here is long after this position that we're trying to quantify. We think this has something to do, well, it's pretty clear it has something to do with the feedback. At this point, the mouse thinks it's in the gain 1 or the gain 2 situation, but then it starts sensing that it's not, and it makes a correction. And so we think this point here is kind of where sensory feedback starts to come into the behavior, and it's long after this, our deceleration point. And to see if that deceleration point has something to do with planning, what we did is we then compared the first and fifth pulls within a condition. If we plot those on top of each other, we see that they start to diverge much earlier. So this is, the mouse is already, in those poles within a condition, it's starting to change its behavior, and that change is happening sometime around this deceleration point. If we look at this across mice, we see that that's pretty systematic, that the feedback comes in somewhere around 75 milliseconds after the mouse starts executing the behavior. And the feed forward delay, or the divergence within a condition, happens much earlier, like 45, 50 milliseconds. So this told us, okay, this is maybe an interesting point to plot here, this deceleration point. So again, this is the position between the home position and the mouth where the mouse decelerates. If we look at that in an individual mouse, in the gain up condition, we see that that position gets closer to the home position, so further from the mouth, as the mouse decelerates sooner. And then when we switch back to the control condition, he then has to increase that again. Okay, and we see that this is happening in an individual mouse in an individual session from trial to trial. It's adapting from pole 1 to pole 5, its behavior. And then when it goes back to the other condition, it has to unadapt its behavior. If we look at the group, so this is now 13 mice. We see that this is really robust, that across mice there's this adaptation from one pole to the next. And it's looking very much like what was observed in humans in the PRISM goggles. So these curves here, the adaptation and the recovery, look very much like mice are in this behavior are doing performing a very similar behavior. They're adapting very quickly. And so now we're excited about this behavior, and there's a lot of different things to do here. And one of the first things we thought to do was to kind of look at how can this behavior be happening in the kind of classical supervised learning that we think is happening in the cerebellum. So in the cerebellum, the classic canonical model is that when a movement happens and there's an error, it doesn't hit the target, there is a climbing fiber signal that signals to the Purkinje cells either that it missed or it didn't miss. And it's kind of hard to see how, without any other information, just it hit the target or it didn't hit the target, how it can always adapt in the right direction and so quickly if it's only getting that signal. And so we thought, okay, let's start looking at that. Let's look at climbing fibers during this behavior. So we use 2-photon imaging, like many people, of dendrites. We're here imaging on the surface of lobule 4-5. We're slicing through a plane of dendrites. So Purkinje cell dendrites are oriented vertically here. We can see the little flashes that if we use a segmentation program to segment this image, it finds, in this particular recording, 307 different regions of interest that look a lot like Purkinje cell dendrites. If we then plot the fluorescence from one of those dendrites, we see that there are these upward deflections, these increases. Those were the flashes that we saw in the video. And we can threshold them and pick out each one of these individual fluorescence peaks using basically a thresholding. It's a little bit more complicated, but basically a thresholding algorithm. And we can pick out what we think are complex spikes. If we plot the frequency of those complex spikes, in red is this one individual Purkinje cell dendrite. We see that the frequency is around 1.5 hertz. All of the dendrites in this particular recording are centered somewhere around 1.5 hertz. And if we look at all the recordings that we've made across all animals, these events are coming at somewhere between 1 and 1.5 events per second, which looks a lot like complex spikes. And other labs, of course, have shown that these fluorescence changes are due to complex spikes. So we think that we can now have a report of complex spiking during the behavior. So the first thing we wanted to do is just look to see what does the lick port do to complex spikes, just the lick port alone. So the way we do that is we record the mouse performing the control behavior. He will pull on the joystick 50 times. So we record all of those 50 pulls, and we record the position that the lick port, the mean position of the lick port during those 50 pulls, and then we play that back to the mouse. We play that exact position back to the mouse. The joystick is down, the mouse can't interact with the lick port here. He's just receiving at a random interval the lick port moving, which you can see in this video at the bottom. So it moves. I was hoping to have these two videos synced so you could see that these two movements of the lick port are basically identical because it's the recording of the mean of the controls. What else did I want to say about that? So now we can now use that. We can now see what happens to Purkinje cell complex spikes when we just give this sensory stimulus. Not too surprising. Other labs have also shown that an unexpected sensory stimulus is going to evoke complex spikes. We see that here. So on the top of these plots, this is the position of the lick port. The gray box is 100 milliseconds following the start of the movement. It'll appear in other figures later. So the lick port moves into in front of the mouse, in front of the mouse's mouth, and we see evoked complex spikes. If we look at the peristimulus time histogram here, we see this big peak in complex spikes. Okay, so now what happens when we have the exact same sensory stimulus, but it's now accompanied by the motor program. So the movement, the mouse is now making this movement. We see that there's no evoked complex spikes here, or very few. We're showing you, some of the best examples here, but in this one, there's very few evoked complex spikes. Okay, and so it looks like at some point, the motor program is coming in and canceling the sensory input that is evoking these complex spikes, or that's one interpretation. So that's kind of the extreme case where we have disconnected the sensory from the motor program. We're playing just the sensory. What happens, do I have the video synced here? No, it's still not synced. Oh wait, so I skipped a thing. So that's one individual recording. This is all of the recordings from a session. We see very little modulation and control, whereas when it's just the sensory input alone, there are these kind of bands of cells which are activated during the sensory input. And they're not activated when that's accompanied by the motor program. If we now look at all recordings, we see that of the cells that were activated with just the movement in control, when it's accompanied by the motor program, there's very little modulation in both an individual, so this particular recording, and then all animals recorded. So just to repeat, it looks like at some point the motor program is canceling out the sensory input.
Brandon?
Yeah.
Hi, Bethany here. Just wanted to ask you a question. Did you evaluate whether this was synchronization of climbing fiber inputs or a triggering of a climbing fiber input? So if you look on average across the data, do you see the same average rate for each ROI?
Not sure I totally understand. So are you wondering if so we clearly see that there's an evoked complex spike here. Are you wondering if there's a complex spike that has disappeared so that the rate?
No, because you talked about cancellation in the control data. But I'm wondering whether it's actual cancellation or whether the spikes that are occurring kind of in random sequences during the control condition are then synchronized during the playback when it's unexpected. So something is triggering olive oscillations, for example, to be synchronous and always release a spike at the same time when you give the playback trigger effectively.
Well, I'm not totally, I mean, so how would you look at that? I mean, so what we're showing here clearly is that there's an increase in complex spiking. You're saying that could be due to a resynchronizing.
If you
I guess
look
we
at
just want to know. I mean, if it's resynchronizing, it's still being evoked.
Yeah, but is it an extra spike or is it synchronization of spikes that would occur anyway because the olive is oscillating?
So an extra spike, you mean, is it, if it wasn't an extra spike, that would mean there was another spike that was subtracted somewhere?
Well, that's what you were just suggesting. In control conditions, you actually suppressed, you called it cancellation of spiking because it's not an unexpected event.
Well, I guess what I meant by that is that we've evoked a spike here, and that evoked spike has been canceled. It's not evoked here.
Okay, yeah, I don't want to make a big deal about it, but it's just a question for me is whether it's so the olive is oscillating all the time, right? And there's probably the output of the olive is both driven by the spontaneous oscillations and synaptic inputs that arrive at the right time during an oscillation to trigger an action potential. And if we think of the olive as a clock, the spike output of the olive may follow those oscillations depending on the state, because the oscillations change their amplitude depending on the state of the olive, on the membrane potential. So you could effectively synchronize olive output by making those oscillations peakier and synchronize, and they would all, if an error signal arrives that's coming from cortex, then maybe all of the olive cells will fire at the same time, or maybe they don't need any input at all, and there's something else that's generating the synchronized spikes just by sharpening the oscillations and making them synchronous. Anyway, I guess what you could do is just look at, very simply, average spike rate over all of those sweeps and see if the average spike rate changes. If it is an extra spike or whether it's a suppressed spike, I don't know.
So the average spike rate here doesn't seem to be very different, and the oscillations in the olive don't seem to be timed to the behavior at all. So this is all random.
Okay.
Right, so here's the baseline rate. We don't really see a difference in baseline rate. What we see is an increase in complex spikes after the behavior. Maybe let's move on and we can discuss that particular point afterwards. Okay, so let's see. So this is the extreme case where the sensory and the motor are completely out of sync, right? There is no motor command. What if we just look at our reaching behavior where it's slightly out of sync? So there's still a motor program, but what the mouse is expecting is slightly different than what it receives. If we look at the complex spike rate in that condition, so in control again, it's fairly stable. On the first pole, when the mouse first encounters this mismatch, that the lick board is moving twice as fast as the joystick, there are some evoked complex spikes here. We see that in kind of this band here. By the fifth pole, that is starting to flatten out. We move to the control condition again, and we now see that there's maybe a suppression of complex spikes here. And by the fifth pole, that starts to flatten out. So it looks like we see something very similar to when we move the joystick alone. We're seeing these evoked complex spikes. But we now saw this suppression that was really surprising to us, that when the lick port doesn't quite make it to the mouth, because the gain has now been decreased, there's kind of a hint that there's a suppression in complex spikes. If we look at an individual mouse, there's a trend here going so these are poles 1 through 5, and this is complex spike rate. There's a trend of decreasing the complex spike rate in the gain up condition or the adaptation condition, and the opposite in the gain down condition. If we now look at all mice that we've recorded from, this is very clear that as the mouse is pulling in the gain up condition, there are more evoked spikes. The dashed line is the baseline spiking rate. We are evoking more complex spikes when it's switched to the faster lick port movement, and with subsequent pulls that decreases. And the opposite for gain down. We have fewer complex spikes than control on the first pull in switching back to control, and that trends back towards the control rate with subsequent pulls. Okay, so there's kind of this hint of a spatial organization here. We see that all of these regions of interest are organized from left to right, and we see there's kind of a pocket of cells that are being activated. Can we get a better sample of that, or better reading of that? So for that, we're plotting just the mean complex spike rate in the first 100 milliseconds in either the first pull in the gain up condition when the lick port is moving fast, or in the first pull in the control when it's moved back to the control condition. We kind of start to see that maybe there's a group of cells here that are activated, and on either side there are cells that are inhibited in the gain up, and that seems to be opposite in the gain down. If we then go back to the same mouse on another day and we look at another region, we see that these cells that were activated, if we look down further down in that same region, we see a continuation of that activated cluster as well as the inhibited clusters. And we can go back day after day and image the same mouse and the same behavior, and we start to put together something that looks like there are bands of activity. So activated in the first pull on gain down is inhibited on the first pull of gain up, and that kind of seems to be alternating across the cerebellum, kind of similar to that image of the rabies staining from Rogrock that I showed at the beginning. So to get a better handle on that, what we did is we just clustered the activity in each individual neuron. So we take each individual neuron and we take the mean peristimulus time histogram from each neuron. Again, 0 is the time when it starts pulling. And we just do k-means clustering on just simply the activity, the mean activity from the pull. When we do that, we see there are basically two types of clusters. There are cells that are activated during the pull and cells that are inhibited during the pull. All of this stuff before the pull is obviously extremely interesting, but for this talk, we're just going to focus on what's happening right after the pull. So in the execution of the pull and not in the planning of the pull. So we see these two different clusters. If we look at what happens when we plot those clusters, just the position of those clusters back onto the regions of interest in the 2-photon recording, we see that there are clusters of cells and they're also spatially organized. So all of the gain up clusters seem to be localized in these bands, and the gain down clusters seem to be localized in separate bands. What happens when we switch back to control conditions? Well, the gain up cluster now is inhibited. The gain down cluster is now activated. And if we look at subsequent pulls, any activation is modulated or dampened by the fifth pull, whether that's activation or inhibition, it gets less by the fifth pull in both clusters. Let's now see let's quantify that. So here we're just kind of looking at the response. Let's take this gray box and take the mean activity of the gray boxes and plot the mean activity of complex spikes versus the lick port position. And The size of these symbols are the pole numbers. So the smaller numbers, or the smaller sizes, are the first pole. The biggest size is the last pole, the fifth pole. And the color is whether this is gain up or gain down. So in gain up, we see that there is a complex spike rate that is evoked. There is a lick port position. With subsequent poles, the lick port position, just like we've shown when we quantified the behavior, is getting closer to the control. Sorry, the control is here. You can see the control better here. This is the control position. So maybe let's look at that one. As in subsequent poles, it's getting closer to the control in terms of complex spikes, but also in terms of the position in both the gain down and in the gain up across clusters. This is just one, so this looks like there is a general effect here. This is just one cluster. This is one recording from one mouse. If we now look at 5 recordings across 4 mice, we see this same trend repeated across all of these mice. The complex spikes are getting fewer with subsequent poles, and the behavior is improving with subsequent poles. Another way of saying that is, let's now plot the behavior. So again, in blue, this is just the behavior adaptation here. Now, with respect to the control behavior, we see that with subsequent poles, it's getting closer to the control behavior, and the complex spike rate in this cluster is getting closer to the control rate. And same for it, so that's in gain up, and the same in gain down. In gain down, as the behavior improves, the complex spike rate also gets closer to control. This is, oh yeah, so this is gain up and gain down, and these are the 2 different clusters. We see the same thing in the 2 clusters. What if we look at all recordings we've ever made? This is now all of the behavior from all recordings, and we see this general trend that the first pole, the smaller one, is producing the highest complex spike rate in the gain up. It's tending towards control with subsequent poles, and in gain down we see the opposite, that's tending towards control. So what this looks like is that the complex spikes are not only encoding whether the mouse missed it went too far when it pulled the joystick, or too not far enough, but also by how much it missed. So here it's missed by a lot. There's a lot of complex spikes. Here it missed by less, and there are fewer complex spikes. And so it looks like these 2 clusters are now not only, when we look at the collective activity in the population of these Purkinje cells, it's not only signaling whether the mouse made an error, but by how much and which direction. And so that now helps to kind of understand how this can happen so quickly. If when the person makes an error, there are a lot of complex spike, or when it makes an error, the complex spike information coming in is telling the cerebellum not only which direction it made the mistake, but it made a big mistake in that direction. Here, on a subsequent throw, it made its mistake still in the same direction, but less of a mistake. That now brings more information into the supervised learning that makes it now maybe possible to understand how it can learn so quickly. So that's the data. The kind of guess on what might be happening here is if we just take the canonical Purkinje cell output, there's a command signal that comes in, activates Purkinje cells, goes through the cerebellar nuclei. Eventually, in this case, this is the first pole in the gain up that produces a pole that is too far, causes inferior olive to produce a lot of complex spikes in a population of Purkinje cells, and some of those Purkinje cells are going to increase, or sorry, this is now an increase in complex spikes, so we're going to have LTD in this scenario. Those synapses are going to get less strong. The next command that comes in, there's less activity coming out of the cerebellar nuclei. This gives us a better performance. It hits the target. fewer inferior olive. We're now at a baseline inferior olive, and there's no change in the signal coming into the Purkinje cells. We now switch to the gain down condition. The command comes in. The signal's now too small. The synapses are too small. We get a smaller change, too small of a movement. Therefore, we now have much fewer activation of the inferior olive. Less activation of the inferior olive is going to cause LTP. And now when the command signal comes in, we're going to hit the target. So that's kind of our guess on one possible result of this observation that populations of Purkinje cells are encoding not only the amplitude, but the sign of the error. I went pretty quickly despite the questions. I would like to thank the people who did the work here. So the bulk of the work was the PhD thesis of Vinh Nguyen, who was just riding his bike. He made it to Côte d'Ivoire after his PhD. So he rode from Paris to Côte d'Ivoire. I saw him in Paris last week, and 2 days ago he set out trying to ride his bike to Kazakhstan. So at the end of that big trip, he's hoping to find a postdoc. The other person involved is Capucine Gros, who's done some of the recent work using pose estimation to look at some of the behavior paradigms. And Luca Misazak, who is a master's student in the lab, who is following up on this using neural pixels. So thanks. Sorry I was a little bit speedy, but maybe that means you're going to have a lot of questions.
Brandon, that was just wonderful. Thank you so much. I think the experiment is ecologically so beautiful, and as you say, it's the animals learn it very quickly, and it's a natural task for them. So congratulations on that really elegant experiment.
Thank you.
So yeah, let's see if we have some questions for Brandon. Just please raise your hand, and yeah, we'll start here. I think. Sasha, I have the question for you.
Yeah. Hi. So the experiments, yeah, like Reza said, the experiment's really beautiful. And the one thing I was curious about is you switch between the gains at 5 trials. That's pretty fast. Do the mice ever anticipate that there's a gain switch incoming, or is that like, or is 5 higher their ability to count and predict?
Yeah, great question. So we don't think they are counting if we, because if we go back to this data here, we don't see any anticipation. So after they've adapted to the gain up condition, then the very next pole is this gain down condition. We don't see much sign that there's been any change in the behavior. They're basically performing the exact same behavior that they performed before. After they adapted, and that's the case here also, right? So this is the same recordings in this 5th pole in the gain down. We now switch to the gain up, and we see that the pole starts out exactly the same as it was in the previous gain. So it doesn't look like there's much anticipation, that there's much counting going on to prepare for the next pole.
Yeah, gotcha. I guess following on that then, do you think there is a trial switch number where the mice would start to like anticipate, or do you think you'd see this behavior even if you just alternated back and forth with single trials?
I have no idea. Really, I mean, is your question like if we did, if we did fewer, I guess if we did more polls, there probably wouldn't be an anticipation. If we did fewer, maybe. Maybe if it was, yeah, maybe. I don't know. I don't know the answer to that.
So Brendan, going back to the concept of anticipation, if they did not anticipate, then you would expect an after effect, meaning that you would expect that following a gain 2 in gain 1 with respect to a block of gain 1s where you didn't change things, so they would start out with an after effect. Lack of an after effect, which I think is what you're showing, could imply anticipation.
So I think I've explained this poorly. I think we do see an after effect. So here, this is, the mouse is going through 5 pulls and gain up. It's adapted down to 0.85. We now switch back to get to the control gain, and he's still performing at the adapted behavior level. And then he has to unadapt that. He unadapts the next pull at the gain up, he's now back. So this is what we think is the after effect. The fact that this one and this one are the same, it's performing the same. You see what I mean, Reza?
I see. I agree with you. Yeah.
But the fact that the pull starts out identical from pull 5 of one condition to pull 1 of the next condition means that he's not really anticipating that. He's pulling. This is why there is that after effect. It's because he's pulling exactly the same as he did in the adapted behavior, but then realizes when the lick port comes in and starts activating whiskers, we presume, then he senses that there's a difference and he says, oh shit, I got to make a correction here.
Yeah. Yeah, I see that. Going to the physiology and the data that you were showing with regard to the banding, so is it correct to interpret that as regions where a particular error is a CS on of the neurons, and that same error is a CS off of some other group of neurons?
Yeah, I think so. I think a CS, you mean by conditioned stimulus or complex spike?
Complex spike.
Yeah, I think that's correct. So I think this band here, the green band, it's being inhibited by the movement. Well, so what we think might be happening is that these groups of Purkinje cells, maybe, I mean, we have to do a lot more experiments, but one guess would be that these groups of Purkinje cells are innervating different types of muscles involved in the behavior. So maybe extensors are green, flexors are brown, something like that. And so then you want to modulate them differently when you're performing, when you want the behavior to, when you want to pull less, you want more flexor and less extensor.
Yeah.
That was maybe a convoluted answer, did I?
Abby has raised her hand. Abby, please go ahead.
Hi, Brendan. Just beautiful talk. I really enjoyed it. I am very interested in whether you think a mouse could learn a mirror reversal of this task and what you might expect with the tuning of these complex spikes, if they could.
Yeah. So you mean we need to get the mouse to push the joystick in order for the lick port to approach?
Right.
So yeah, we absolutely want to do this. We haven't started it yet, and I think they will do it. I mean, I think it's not, every mouse knows in its cage how to reach out, grab something, and bring it to its mouth. So it's not a surprise they can do this behavior. Reaching out and moving something away, causing something to come closer to its mouth, it's going to be a little bit more of a stretch, but I think with time we're going to be able to get it to do that. And then, so I think we'll see, like in the basic control experiment, we're going to see the exact same thing we see here. But what would be interesting is to switch that mirror in one session. Right? That's going to be a Herculean experiment because we're going to have to get it to do a lot of pulls to do that. And in that case,
maybe a question is, why do you think the adaptation to that would be so much slower than this within direction adaptation that you're seeing? Like, do you have a mechanistic insight to why you think that would be harder for the animal? Because I agree with you. But the intuition about the sort of mechanism behind why it's so much harder, I think, is very interesting.
So one guess would be that, to decrease it, to decrease, how to say this? So the control behavior is just pulling something to its mouth, pulling it a little bit more, pulling it a little bit less. This is just changing, fewer synapses on that given set of Purkinje cells, just kind of titrating the number of synapses that are involved with that. To do the opposite behavior, you basically have to flip the sign of all of those synapses, right? The ones that are inhibited and the ones that are excited have to do the opposite thing. And so maybe that would take a lot longer to do. There's kind of hints of this, so we learn, with Prism goggles, we learn this very quickly, like it was shown by Martin et al. But if you wear Prism goggles that flip the world upside down and do things completely opposite, this takes a lot longer to adapt to. And so my guess is maybe it's not just nudging the system a little bit in one direction or the other, it's completely asking it to do the opposite thing, and it just takes more, changing more synapses. That's a guess. I don't know, Abby, but that's my best guess.
No, that's great. That's great. Reza, do I have time for one more question? May I?
Let me go to Yoshiko,
Yeah,
and then
for
I'll.
sure.
Okay,
No,
Yoshiko,
go
please.
ahead, Abby. Yeah, we still have 12 minutes.
Hi.
Thank you, though.
Well,
I didn't understand the question.
Yoshiko, go ahead and ask your
Okay.
question.
Can you
Go to
hear?
Ab. Yeah, go ahead.
Okay, so I'm wondering, if you compare the same, so I'm asking the last part, the amplitude part. Sorry, I'm late, so I might just simply miss. But so if you compare the same amplitude error at beginning of the session and the later part of the session, whether the complex spike activity is different or not. Because the reason I'm asking this is because by the way, I have been studying saccade adaptation, and when we keep the error amplitude same through the entire adaptation session, the basal ganglia activity changes, and then the colliculus activity change, and colliculus send the error signal to the cerebellum through the inferior rib. And even if we keep the amplitude of the error same, the colliculus activity change and the basal ganglia activity change. So the climbing fiber complex spike activity change during the adaptation session is not simply the amplitude change, the error size change during the adaptation. It may more animal get tired by repeating the trial kind of thing also contributes. So I'm curious whether if you compare the beginning and end of the session, same amplitude of the error, you see the complex spike activity change or not, different or not.
So we have done the experiment where we look at the behavior in the first, 100 polls and compare, or the first 100 gain changes, or first 100 polls, and compare that to the last 100 polls of the session. We've done it for the behavior, and we see the behavior is identical. So it doesn't learn any different at the beginning of the session versus the end of the session. I'm not sure we have looked at the complex spiking. That's a good point. So I think we'll have a look at that.
Okay, thank you.
Abby, back to you.
Thanks. So I was also curious about the complex spiking. You proposed this nice model of extensor and flexor modules being differentially activated. And I'm wondering if you have looked at sort of trial by trial comparisons of the up and down modulated CSs. That is, you've nicely grouped this by trials 1 through 5, but I'm wondering about sort of internally whether maybe on trial 1, since you have so many switches, whether you've looked at the sort of co-probability of those two up and down modules as predicting the amplitude of the behavioral change on that next trial, if that makes sense, or if one, say the upbound is dominating or the downbound is dominating. Have you looked at that?
We have. You've put your finger exactly on the issue we have, which is that, so if we look at exactly that, so we look at either the response of the cluster, of one cluster, the other cluster, difference between the clusters, and then look at the performance on the very next behavior. So there's a little bit of an issue here, which is the next pole. So if we do that, we see a beautiful correlation that there's the next pole is affected by the amplitude of the response, either difference in clusters, one cluster, the other cluster, beautiful correlation from pole to pole. But it's a little bit circular because the second pole is always smaller than the first pole, and the calcium response is always smaller than the first pole. So it's not surprising that we've seen this, this correlation from pole to pole. We've done many different attempts to try to normalize that, looking at the difference from one pole to the next pole, given that it's a pole 1 or a pole 2 or a pole 3, and looking at the complex spike of that. And we haven't been able to tease apart any pole-to-pole changes predicted by the complex spike rate. And so that raises another issue. So I've shown this kind of simplified model here where the command is going through the Purkinje cell to produce the behavior. But that's obviously a major simplification here. What could be happening also is that part of it is going through the cerebellum, part of it is going down to the motor neurons without going through the cerebellum. And it's not entirely a forward model. There could be some inverse model here where the cerebellar nuclei are changing the command. And the changed command from pole to pole is an effect of whatever plasticity happened here to create this comparison, but is not necessarily dependent on pole to pole plasticity. I think I explained what I wanted to explain, but I'm not sure that's totally clear. I mean, the upshot is that we see that correlation from one pole to the next, but I think it's really a circular argument. And all of our attempts to normalize that out and make it not circular have failed so far. I would be very interested in hearing your thoughts about that if you had any ideas. It doesn't have to be now.
Cool. I'll reach out.
Great.
Wonderful. Are there any other questions for Brandon? Well, Brandon, thank you so much. This was wonderful.
Thank you all. It was a pleasure to do. I'm really happy to have these seminar series.
Wonderful. All right. Have a wonderful day. Bye, everyone.
Goodbye, everybody.